Sofosbuvir and Ribavirin. Pharmacology treatment. What is the Sofosbuvir? Sofosbuvir, brand name Sofosbuvir, is a drug used to treat hepatitis C infection. In combination with other therapies, Sofosbuvir can effectively cure hepatitis in 90% of patients. It inhibits the RNA polymerase that the Hepatitis C Virus HCV uses to replicate its RNA. It was discovered at Pharmacologic and developed by Gilead Sciences. We can see a video.
Hi, I'm Dr. Joe Galati. We're going to be talking about some really great news about hepatitis C. So here you are, a typical baby boomer born between the years 1945 and 1965, going about life without a care in the world. And it's been recommended that all baby boomers get tested for hepatitis C because of the increased risk in this group. So you do as you're told, only to find out that you indeed have hepatitis C. This is always a shock to your world. There is increased concern for yourself and your family. The life as you know it is in question. Thinking you've done well. The family cat, nice cars. You've saved for the future and been able to buy nice stuff. But now you feel as if you've been struck by lightning. Everyone always wonders, why me? Why did this happen to me? So what exactly is hepatitis C? Well, hepatitis C is a disease of the liver. The liver is involved in almost 200 different processes to keep us all healthy. The hepatitis C virus invades your liver, attacks it, and potentially makes you very sick. You may feel absolutely 100% normal at first, but most have some mild fatigue. They may have some very vague flu-like symptoms, and in about 15 to 20% of the people with hepatitis C, they develop cirrhosis. Once cirrhosis develops, you can develop liver cancer, and in some situations, you may need a liver transplant. Some die from hepatitis C. Despite all of this, we do have smiles on our face, and the future looks very bright for those with hepatitis C. After years of researching new drugs to treat and eradicate hepatitis C, the time has come, with music in our ears, joyous that we have new drugs to treat hepatitis C effectively, safely, and with far less side effects than we've been used to. Here in America and in other parts of the world, hepatitis C patients should be celebrating, rejoicing that we now have a green light with all the regulatory work done with FDA approval of 2 new, very effective and very safe drugs for hepatitis C. The 2 new drugs are Sofosbuvir and Ribavirin, and they have recently been approved for use, and the success rate is absolutely staggering. These new drugs can attain an 80 to 90% cure rate in a wide range of patients with hepatitis C.
Medical uses. Sofosbuvir is used for the treatment of chronic hepatitis C, genotype 1, 2, 3, and 4 in combination with pegylated interferon and ribavirin or with ribavirin alone. It is also used in HCV patients with an HIV co-infection. The treatment is based on a number of clinical trials, for example, the Electron trial, which showed that a dual interferon-free regimen, Sofosbuvir plus Ribavirin, produced a 24-week post-treatment sustained biological response. SBR24, rated of 100% for previously untreated patients with HCV genotype 2 or 3. Let's start the second video.
The hepatitis C virus, HCV, is one of the leading causes of chronic liver disease and is a leading indication for liver transplant. The current standard of care for the treatment of genotype 1 chronic HCV infection is a triple drug regimen of pegylated interferon plus ribavirin plus a protease inhibitor. Although effective in 68 to 75% of treatment-naive patients, the tolerability and dosing schedules of currently available treatment regimens may restrict use in some patients. The development of safer and/or more effective investigational compounds is ongoing. Our increasing understanding of the HCV life cycle has revealed a number of potential targets for pharmacologic intervention. The hepatitis C virus, a positive single-strand RNA virus, enters the body via the bloodstream and targets the hepatocytes. The virus binds to the hepatocyte via a specific interaction between the viral structural proteins and liver cell-specific receptors on the hepatocyte cell surface. The virus then enters the hepatocyte via receptor-mediated endocytosis. Once inside the hepatocyte, the viral membrane fuses with the endosomal membrane, releasing the viral RNA into the cell cytoplasm. The viral RNA then travels through the cytoplasm to the endoplasmic reticulum, where it is translated by host ribosomes, resulting in the formation of the hepatitis C polyprotein within the endoplasmic reticulum membrane. The resulting HCV polyprotein consists of 10 structural and non-structural proteins that play essential roles in the virus life cycle and represent potential targets for pharmacologic intervention. The NS3 and NS5B protein have thus been key targets for antiviral drug development. NS3 consists of a C-terminal helicase domain and an N-terminal serine protease domain, which together with NS4A is responsible for the cleavage of all downstream non-structural proteins, an essential step in the HCV replication process. The non-structural proteins, together with host proteins, form the replication complex that facilitates the production of new viral RNA. The newly formed RNA is then packaged and assembled into new viral particles, which are then released from the cell to go on to infect other hepatocytes. Agents being developed for use in the treatment of chronic hepatitis C include compounds that inhibit NS3 protease. Feldaprevir is an investigational NS3 protease inhibitor that has a linear tripeptide structure with a C-terminal carboxylic acid group, which forms a non-covalent complex with the active site of NS3 protease. This in turn prevents the cleavage of the HCV polyprotein, and thus the release of the non-structural proteins necessary for the production of viral RNA, resulting in a reduction in viral replication. Phase 2 studies with feldaprevir as a part of combination therapy with pegylated interferon and ribavirin have been completed. Phase 3 studies are nearing completion. The second non-structural protein that has received much attention as a potential target for pharmacologic intervention is the NS5B polymerase. The NS5B polymerase is the catalytic center of the HCV replication complex, where it is directly responsible for RNA synthesis. Like other viral polymerases, NS5B has the right-hand structure with palm, finger, and thumb domains. There are at least 4 allosteric binding sites on the polymerase, namely thumb pockets 1 and 2 and palm sites 1 and 2. The investigational agent BI207127 is a non-nucleoside thumb pocket 1 inhibitor. By binding to the thumb pocket 1 site of NS5B polymerase, BI207127 blocks the conformational changes to the enzyme that are necessary for RNA synthesis. With their complementary mechanisms of action, BI207127, in combination with Faldaprevir and Ribavirin, is being studied as part of an interferon-free regimen. Phase 3 studies investigating the safety and efficacy of this regimen are currently initiating with the goal of delivering an interferon-free treatment to patients with chronic hepatitis C.
Adverse effects. As Sofosbuvir is always combined with other drugs such as Ribavirin and interferon, only the adverse effects of these combinations have been evaluated. Common side effects are fatigue, headache, nausea, rash, irritability. Most side effects are significantly more common in interferon-containing regimens as compared to interferon-free ones. For example, fatigue and headache are nearly cut in half. Influenza-like symptoms are reduced to 3 to 6% as compared to 16 to 18%. Neutropenia is almost absent in interferon-free treatment. Let's start the video. Mechanisms of action. Sofosbuvir is a product that is metabolized to the active antiviral agent 2-deoxyfluorocy-methyluridyl triphosphate. The triphosphate serves as a synthetic substrate for the NS5B protein, which is the viral RNA polymerase, and that acts as an inhibitor of viral RNA synthesis. Ribavirin. Ribavirin is a one-side ribonucleate analog used to stop viral RNA synthesis and viral mRNA capping. Thus, it is a nucleoside inhibitor. Its brand names include Copagus, Ribavirin, Ribavirin, Vilona, and Virasol, and is an antiviral drug used off-label for severe RSV infection individually, notably for persistent hepatitis C infection. Can be used in conjunction with peginterferon alpha 2b or peginterferon alpha 2a and some other viral infections. Let's show another video about hepatitis C.
Hello, I'm Dr. Gregory T. Everson. I am a professor of medicine, director of hepatology at the University of Colorado, Denver. And I am the lead author of a paper looking at a promising all-oral investigational treatment for patients with genotype 1 hepatitis C infection. We conducted this phase 2A open-label study to determine whether the all-oral combination of 3 direct-acting antiviral agents, Declatosvir, Isoniprevir, and BMS 791-325, could be used safely and effectively in previously untreated patients with genotype 1 hepatitis C infection. Now, there are 6 strains called genotypes of hepatitis C. The most common in the United States, Europe, Japan, and China is genotype 1. This genotype represents 75% of the hepatitis C infections in these countries. Hepatitis C genotype 1 has been the most difficult to treat, requiring up to 1 year of interferon-based therapy. The overall effectiveness of current treatments has been limited, however, by side effects and intolerability of interferon, ribavirin, and the first-generation direct-acting antivirals telaprevir and boceprevir. Clearly, interferon-free, ribavirin-free treatments are desirable. We use a term, sustained virologic response, or SVR, to measure the success of anti-hepatitis C therapy. A patient with undetectable hepatitis C RNA in blood samples obtained 12 weeks after the end of a course of therapy has achieved SVR and is cured of hepatitis C. Comparative rates of SVR with current FDA-approved therapies in patients with hepatitis C genotype 1 have been 40% with peginterferon ribavirin and 70% with peginterferon ribavirin plus either telaprevir or boceprevir. But again, because of the interferon ribavirin backbone and the side effects of telaprevir and boceprevir, this treatment has been hampered by side effects, adverse events, complicated treatment regimens, and long durations of treatment. In many cases, patients required 48 weeks or more of therapy. 2 new drugs, Sofosbuvir and Sofosbuvir, in combination with peginterferon and ribavirin, have just received FDA approval for treatment of hepatitis C genotype 1. These treatments are more tolerable, associated with fewer side effects, administered as a shorter course, in the case of Sofosbuvir, only 12 weeks. And they promise to enhance the rates of SVR. Nonetheless, they are still administered with peginterferon ribavirin in the patients with hepatitis C genotype 1. Now, we are in a very exciting era of hepatitis C research that is poised to change the current treatment paradigm, with several all-oral regimens being studied that are interferon-free and ribavirin-free and with shorter treatment durations. The question we addressed in our Phase 2A study was whether patients can achieve high SVR with the interferon and ribavirin-free regimen of Declatosvir, Isoniprevir, and BMS 791-325. In our study, 66 treatment-naive, non-cirrhotic patients infected with genotype 1, predominantly genotype 1A, were studied. The primary endpoint of the study was achieving hepatitis C RNA levels of less than 25 international units per milliliter at 12 weeks after treatment, or SVR 12. Patients were randomly assigned to 4 groups. All received Decladisvir 60 mg once daily and Asunoprovir 200 mg twice daily. Patients were randomized to either 75 or 150 mg of BMS 791325 twice daily for either 12 or 24 weeks. The results from this trial were very promising. This regimen, regardless of dose of BMS 791325 or duration, was highly effective in achieving rapid viral reduction and SVR. In 64 study patients, hepatitis C RNA was less than 25 international units per milliliter by week 4 of treatment, and rates of SVR were 90% or more in all groups. 2 patients experienced viral breakthrough and 1 patient relapsed. The treatment was generally well tolerated. There were no grade 3 or 4 increases in levels of alanine or aspartate aminotransferase or bilirubin. There were no patient deaths and no patients discontinued the study as a result of serious adverse events or adverse events related to the 3-drug treatment regimen. The most common adverse events were headache, weakness, and gastrointestinal symptoms. We conclude that this is a promising interferon and ribavirin-free all-oral regimen that warrants additional study in the treatment of chronic hepatitis C. This regimen achieved high rates of SVR and was generally well tolerated. Phase 3 studies with a fixed dose combination of Decladisvir, Asunoprovir, and BMS 791325 are underway and include treatment-naive, treatment-experienced, and cirrhotic patients. Thank you for your attention.
Medical uses. Ribavirin is used primarily to treat hepatitis C and viral hemorrhagic fevers, which is an orphan indication in most countries. In this former indication, the oral capsule or table form of ribavirin is used in combination with pegylated interferon, including in people co-infected with hepatitis B, HIV, and in the pediatric population.